Jurors, in light of our late start today, I'm going to keep on. Everybody good? Thumbs up. You may call your next witness.
Commonwealth will call Bryce Raymond.
Do you solemnly swear or affirm the testimony you'll give in the cause now and hearing will be the truth, the whole truth, and nothing but the truth, so help you God?
I do.
Thank you, sir. You can be seated.
This way, sir. You just watch your step for me.
You may proceed.
Thank you very much, Your Honor. Good morning.
Good morning.
Could you please state your name for the record and spell your last name?
Bryce Raymond, Raymond.
Could you please tell...
Bryce, Bryce.
Could you please tell us your educational background?
I have a Bachelor's of Science in Forensic Science from Penn State University and a Master's of Science in Biology from Tufts University.
And what is your occupation, sir?
I'm currently a Forensic Scientist 2 in the DNA unit at the Massachusetts State Police Crime Lab.
And how long have you been employed at the Massachusetts State Police Crime Lab?
Since June of 2019.
And can you please tell the jury your present duties in the DNA unit and the nature of the work that you do?
Yes, as a Forensic Scientist 2, I am in charge with processing evidentiary items through our DNA lab processing, as well as interpreting and making any conclusions to known standards.
Do you have any specialized training to be a DNA analyst?
Yes, I received at least 6 months of specialized training in which I read all the protocols and procedures at the lab. I attended lectures and took quizzes, as well as worked on mock evidentiary items through the lab processing. And at the end of testing, I take a competency test.
And did you pass that competency test?
Yes, I did.
So does your testing and training stop after that initial 6-month period?
No, every year we have to do at least 8 hours of continuing education in order to stay knowledgeable in the field.
And do you continue to be tested at the lab?
Yes, we do.
And what kind of testing is that called?
That's called a proficiency test.
And how does proficiency testing work at the lab?
Yeah, so twice a year we have to take a proficiency test to continuously show that we have the skills and knowledge in order to perform DNA analysis. This is given by an outside accrediting body, and when we complete our work, we send in the results to that accrediting body for them to grade.
And what are the results of your proficiency testing?
I have passed every one I have taken.
So approximately how many samples have you had the opportunity to analyze for DNA testing?
Approximately 2,000.
And have you testified before in Massachusetts about DNA?
Yes, I have.
Does the Massachusetts DNA unit at your lab follow standardized protocols and procedures?
Yes, we do.
And who sets those protocols and procedures?
They are based on some accrediting bodies, such as the FBI, as well as validations that are done at the lab.
Well, you anticipated my next question, which is your lab, the Massachusetts State Police Crime Lab, accredited?
Yes, we are. We are accredited by ANAB, which is the ANSI National Accreditation Board, as well as the FBI Quality Assurance Standards for DNA laboratories.
And what does accredited actually mean?
That means we are following a specific set of standards to make sure that the scientific work that we are performing is accurate and reliable.
And is that a national accreditation?
Yes, it is.
And does your work undergo any kind of review process?
Yes, all work goes under 2 different types of review processes, a technical review and an administrative review.
And is that review process required under the accreditation standard?
Yes, it is.
And so if you could just explain technical and administrative review, please.
Yes, a technical review is one that is done by a qualified analyst that takes a look at your work to make sure that you followed all the proper protocols and procedures and made sure that any conclusions you made are scientifically sound. An administrative review looks over the file to make sure there's no spelling mistakes, no grammar mistakes, all the pages are numbered and initialed, and other administrative things such as that.
And why is that all important?
That's important to make sure that we provide the best quality work that we can.
I now want to talk about DNA. What is DNA?
DNA stands for deoxyribonucleic acid. It's the genetic blueprint of life and what makes you, you. You inherit your DNA from your parents, so you get half of your DNA from your mom and half of your DNA from your dad. And no two individuals have the same DNA profile except for identical twins.
Is DNA the same in every cell of a person's body?
Yes, so whether we test blood, saliva, hair, skin, we should expect the same DNA profile if they came from the same person.
So what type of biological material can DNA be found in?
So that can be blood, saliva, skin, hair, sweat, urine, anything biological.
So can you explain generally the process that is used for forensic DNA analysis?
Yes, at the lab we have a 4-step process. The first step is extraction. This is when we break open the cells and collect the DNA. The second step is quantitation or quantification. This is when we can get an estimate of how much human DNA is present as well as how much male DNA is present. And that's so we can target an appropriate amount for the third step. And that third step is amplification. This is when we make millions and millions of copies of specific areas on the DNA and are fluorescently tagged. And then the fourth step is separation and detection, also known as capillary electrophoresis. This is when we put the DNA on an instrument that separates out the DNA by size and has a camera to detect those fluorescently tags. And that produces raw data.
And what is the goal of that raw data?
The goal of that raw data is to then be read by an interpreting analyst in order to interpret the DNA profile.
And what is a DNA profile?
A DNA profile, so the specific areas that we are copying during amplification is called STRs or short tandem repeats. That is just areas on the DNA that repeat different lengths for a person. So one person could have 5 repeats, another person could have 6. And it's the combination of all those numbers is what makes up a DNA profile.
So how many locations are you testing?
We test 26 locations as well as a sex determining location.
And what is batch testing?
Batch testing is what we do at the lab and is a common practice in forensic science in which if many items need to be processed for the same lab step, one analyst will process all those items at once on the same instrument.
So why do you use batch testing?
We do that for efficiency, as it is better for one analyst to process, say, 50 items at one time on one instrument than 50 analysts trying to process one item each on the same instrument, one after each other.
Is there a protocol that you follow prior to retrieving any evidence to prepare your lab area to avoid contamination?
Yes.
So how do you avoid contamination?
To avoid contamination, I first put on protective personal equipment, and that can include a lab coat, a mask, a hairnet, and for me, a beard net. When I go to my lab bench, I also put on gloves, and I spray the lab bench with bleach, clean that off, and lay down lab bench paper before I proceed with any testing.
So why do you use bleach?
The bleach is to destroy any DNA that may be present on the lab bench.
And why do you use bench paper?
We use bench paper to prevent any contamination from items that I may be testing to be deposited onto the lab bench for the next person.
And why are you wearing PPE?
The The PPE is also to prevent any DNA that I may touch to go over to another lab bench or any of my DNA to be deposited on the items.
And once your area is prepared, can you please tell us what you first did and what you first do with items that you're going to sample?
Yes, so for sample preparation, I get a list of items that have been selected for DNA analysis. I get those items out of storage in our secured walk-in room. I take that container of evidence back to the lab bench. I scan it into my custody and take a photocopy. I then open up the container, grab the items that I need, and put those aside ready for testing. I seal up the container with my initials and date and put the container away.
And did you work on lab case 23-00596?
Yes, I did.
And what did you initially do to prepare the samples for DNA analysis?
I did sample preparations on some items.
Okay, so I'm going to ask you specifically about samples. Did you prepare samples 5-1.1, sample from swabs of red-brown stained cave on basement floor? 7-10.3.1, sample from the stained area C on the blade of the hacksaw. 7-10.4.1, sample from the handle of the hacksaw. 9-22.1.1, sample from stain A on white towel number 1 with red-brown stains. 9-28.1.1, sample from stained area A on apparent hairs. 9-31.1, sample from cutting of red-brown stain from piece of rug from bag number 5 from 300 Forest Street. Did you prepare samples from those?
Yes, I did.
So after preparing the samples, what did you next do?
After preparing the samples, I then did the first lab step of extraction.
And how did you do the extraction step?
Yes, to each of the tubes, along with the controls, I added chemicals to the tubes and put them in an oven, and that is done to break open the cells in order to free up the DNA. I then take those tubes and put them on an instrument that separates out the DNA and puts them in a separate tube in liquid form, and that tube is called an extract.
Did you use equipment?
Yes, I did.
What equipment did you use?
I used an oven, and the instrument was an Easy One robot.
And how did you use the equipment?
For the oven, I just placed the samples in and took them out. For the Easy One robot, I put the samples on the robot and then programmed the robot with the proper protocol to use for the extraction and collection, and then got the tubes off of the robot when the process was done.
And I think you said that that is now called, what is that now called?
That is now called an extract.
Okay. And then what did you do next?
Next, I took a small portion of the extract, known as an aliquot, and put that into a separate tube and prepared that for the second step of quantification.
So now we're on step number 2?
Yes.
Okay. And what items did you do quantification for?
The same items that were previously mentioned.
And how did you perform the quantification step?
Yes, so for each of the aliquots, I put a portion onto a 96 well plate along with other chemicals. I then placed that 96 well plate onto an instrument, and that instrument does a series of heating and cooling, and that produces the data that gives us the estimation of DNA.
So I believe you just named the equipment that you used.
Yes, it is a real-time PCR system.
Okay. And so how did you use that equipment?
So I put the 96 well plate onto the instrument. I then used the computer program associated with it to say that to start the quantification process. And then after it was done, I collected the data off of the equipment and then put it on our computer drive.
And then what did you do next?
After that, I discussed the quantitation results with the interpreting analyst of this case, who is Salmon Salem.
And then what did you do?
Then we came up with how to do amplification strategies for each of the items. And then once that was decided, I went forward with the third step of amplification.
So step number 3 is amplification.
Correct.
And what items did you perform amplification for
Those same 6 items.
that we previously named?
Correct.
Okay. And how did you perform the amplification step?
Yes. For each of those items, I got the extracts, and I either used all of the extract or a portion of the extract to target an appropriate amount of DNA for testing.
Let me just stop you for a second. What's extract?
Extract was the product from the first step of extraction.
Okay. And now finish your answer. I'm sorry to interrupt you.
Yes. So for those samples, I added chemicals and put them into an instrument known as a thermocycler. And that also does a series of heating and cooling, which makes the copies of the DNA that are fluorescently tagged. And at the end of that process, the tube is now known as an amplicon.
And then what did you do next?
Next, after the amplicons were done, I then proceeded on to the fourth step of separation and detection.
And which items did you do the fourth step for?
The same 6 items that were mentioned.
So you did all the steps of DNA for those 6 items.
That's correct.
And how did you perform the detection step?
Yes. Of the amplicons, I took a small portion of each amplicon and put it on a 96 well plate, added chemicals to the plate. I then put that plate onto an instrument known as a 3500 genetic analyzer. I then programmed the genetic analyzer to perform the separation and detection. And then the instrument separated out the DNA fragments by size and with the camera detected those fluorescent tags.
And then what happens next?
After that data is generated, I take the data off of the instrument and put it on our computer drive.
And then what happens?
After I'm done, I let the interpreting analyst know that data has been collected, and I hand over any worksheets that I generated to them.
And who does the interpretation?
In this case, it was Salmon Salem.
So you did not do the interpretation here?
I did not.
I might have one moment, Your Honor.
Nothing further, Your Honor.
Cross-examination.
Just a few questions. Mr. Rader. Good morning.
Good morning.
And you've taken a lot of proficiency tests. You've passed them all, correct?
Yes.
You take pride in what you do, don't you?
Yes.
And you're running these machines, the analyzer and everything else, and you're running them consistent with the protocols that are required to be followed to run them accurately, correct?
That is correct.
And what you do is you depend on the fact that the samples that you get for testing are samples that will result in reliable results, correct?
Correct.
But what is your title at the lab?
I am a forensic scientist too.
Okay. And are there forensic scientists 3 and stuff like that? Forensic scientist 1?
Yes.
Okay, but you're a 2, correct?
Correct.
And with your knowledge and experience, it's fair to say, isn't it, that you have no idea where the samples that you subjected to DNA testing came from, correct?
That is correct.
And in fact, you have no idea how they were collected, wherever they were collected from, correct?
That is correct.
And in fact, you can tell the jury that at times when you conduct testing like you just described, the results will indicate that there are mixtures of different DNA in a particular sample, correct?
That is correct.
And whenever you get the results that result in a mixture, you can't determine, as a forensic scientist 2, or for that matter, forensic scientist 3, you can't tell how those 2 different substances came into contact with each other, resulting in a mixture, correct?
Our testing does not determine how DNA got onto an item.
And hopefully it's a simple question. Your DNA results do not tell you anything about what happened at a, let's just call it a crime scene, what happened at a particular scene or where they were collected, correct?
Correct.
Thank you, Steve.
Any redirect?
Thank you.
You may proceed.
Thank you very much, Your Honor. Good morning. Can you please state your name for the court and spell your first and last name?
Sure. My name is Brianna Kiesel, B-R-I-A-N-N-A. Last name Kiesel, K-I-E-S-E-L.
And what is your educational background, ma'am?
I have a bachelor's in biology from Arcadia University and a master's in forensic science from Towson University.
And what is your occupation?
Currently, I'm a forensic scientist too with the Massachusetts State Police Crime Lab,
And what unit are you assigned to?
the DNA unit.
And how long have you been employed at the Massachusetts State Police Crime Lab?
6 years.
And what are your responsibilities as a forensic scientist too?
I am authorized in processing, interpreting, and reporting samples that were collected from crime scenes, as well as standards collected directly from an individual. I can also technically review other analysts' work when it concerns batch files, which we call files that have standards collected directly from an individual.
And do you have training for this position?
Yes, I do.
Can you tell me about your training?
Sure. Our training is a minimum of 6 months, and during that time we have lectures, quizzes, and this all culminates into a final written exam. We also have a practical component in the laboratory where we take several steps for sample sets forward, and that all culminates into a final written exam, as I mentioned, and a final lab practical.
And did you pass that written exam?
Yes, I did.
And that's called a competency evaluation?
Yes, that is a competency, the lab portion of it.
And do you also have to take proficiency testing?
Yes, we do.
And can you explain that to the jury?
A proficiency test is an ongoing competency test where we have to take samples forward twice a year to prove that we are that we are knowledgeable and still performing that skill or method according to laboratory standard operating procedures.
And have you passed your proficiency testing?
I have, yes.
And have you testified in court about DNA?
I have.
So I want to now talk to you about this case, 23-00596. Prior to retrieving any evidence in 23-00596, how did you prepare to process the evidence?
So once I was assigned the samples, I cleaned my bench space with bleach and wiped everything down, every instrument that I was using. And once that was done, I laid down a piece of fresh bench paper.
And directing you now to lab number 23-00596, did you work on items 9-27.2.1, the sample from the interior of the dark gray slippers from bag 4 of 300 Forest Street, and item 9-27.3.1, stain A of the exterior left gray slipper?
Yes, I did process those samples.
And what work did you do on those two items for sample prep?
For sample prep, they were already prepared by our criminalistics unit, so I just went into a secure freezer area to retrieve the packaging. I removed the tubes from the packaging and just visually examined the items within the tube.
Did you also work on item 9-32.1.1, the sample from the interior sleeve cuff of the Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of the Tyvek suit, and sample from stain D on the interior right sleeve of the Tyvek suit?
I did, yes.
And what work did you do for that?
Those samples I performed just like I previously mentioned, where I went to the secure freezer area, retrieved the packaging, removed the tubes from the packaging, and just observed the samples within the tube.
So you also just did the sample prep for that?
Yes.
Did you also work on item 16-13.1, the unknown tissue recovered from the Swampscott dumpster?
Yes, I did.
And what work did you do on this item?
So in this case, this item was still in a specimen jar, so I retrieved the packaging just like I previously mentioned, made sure the seal was secure as I did with the other samples. And then after that, I opened up the jar and sampled a few items from the jar and placed it into a tube for processing.
So you actually had to make the sample for this?
I did, yes.
Okay. And how many samples did you make?
So this was 16-13.1. There were two tubes we referred to as 16-13.1A and 16-13.1B.
And did you do any further work?
No, that is where my work ended.
Okay. And where did the samples then go?
I then passed the samples off to Madison Frank for extraction.
Cross-exam. No question. The witness may step down.
Thank you.
You may proceed.
Thank you very much, Your Honor. Can you please state your name for the court, spelling your name?
My name is Madison Frank. That is MADISON FRANK.
And what is your educational background, ma'am?
I have a Bachelor's of Science degree in Forensic and Investigative Sciences from West Virginia University.
And what is your occupation?
I'm a forensic scientist too in the DNA unit of the Massachusetts State Police Crime Lab.
And how long have you been employed at the Massachusetts State Police Crime Lab?
I've been employed there since October of 2019.
And what are your responsibilities?
As a forensic scientist too, I will perform DNA analysis on either known or evidentiary biological samples, interpret those results, and report out my results in a case file.
And have you had training for this position?
Yes, I have.
And have you passed your proficiency testing?
Yes, I have.
And have you passed the competency testing that was part of your training?
Yes, I have.
And have you testified in court about DNA?
Yes, I have.
Directing your attention to lab number 23-00596, did you do work on this case?
Yes, I did.
I want to ask you now about items 9-27.2.1, the sample from the interior of the dark gray slippers from bag 4 of 300 Forest Street, 9-27.3.1, the stain A on the exterior left gray slipper, 9-32.1.1, the sample from the interior sleeve cuff of the Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of the Tyvek suit, 9-32.5.1, sample from stain D on the interior right sleeve of the Tyvek suit, and 16.13.1, the unknown tissue recovered from the Swampscott dumpster. I believe I just gave you 6 of those samples. Did I mention 9-32.2.1, the sample from stain A on the exterior leg pant of the Tyvek suit? So those would be the 6. What work did you do on those?
DNA analysis is a 4-step process, so I perform steps 1 and 3 of that process on those samples.
Okay. And what are 1 and 3?
Step 1 is extraction, and step 3 is amplification.
Okay. And how did you do step 1, the extraction step?
For extraction, I received the samples and I added chemicals to the samples and the controls and then put them in an oven so that we can break open the cells to obtain the DNA. Then those samples will go on an instrument that helps to clean up the sample so that we're only dealing with the DNA and no other cell parts. Then after the extracts come off of the instrument, then I will take a small portion of that sample and I will put it in a new tube to be used for the second step.
And which instrument did you use or equipment?
An Easy One robotic instrument.
Okay. And how do you use that instrument?
The samples will go onto the instrument in tubes, and then there will be new tubes where the samples will go through processing in the instrument to put out just the liquid and DNA portions of the sample.
And then what is that biological material then referred to?
An extract.
And what is that extract then ready for?
The extract is then ready to be quantified in the second step.
So did you do that second step quantification?
No, I did not.
Who did that second step for those 6 items?
Troy Adams did the second step of quantification.
And why is that?
We use a batching process when we are processing our samples so that it is a more efficient and streamlined processing.
And are you qualified to do all of the steps of DNA, the 4 steps that you described?
Yes, I am.
Okay. And then did you do more work on this case with the 6 samples?
Yes, I also did the third step of amplification.
On all 6 of those samples?
Yes.
And that being 9-27.2.1, 9-27.3.1, 9-32.1.1, 9-32.2.1, 9-32.5.1, and 16-13.1. Did I get that right?
Yes, that is correct.
Okay. So you did the third step, and how did you do the amplification step?
So I took the extracts that were generated from the first step, and I took portions of those samples and added chemicals to the tubes with the samples, and then I put them onto an instrument called a thermocycler, and this will have the samples go through periods of heating and cooling, and this is to make millions of copies of the specific DNA that we are looking for to be able to visualize it in further down in our processing.
And which equipment did you use for that?
A thermocycler.
Okay. And then how do you use that equipment?
The samples just go on, they are in small tubes, and they will go on into small holes in the thermocycler, and then it is a metal plate that they are in, and it will go through different steps of heating and cooling.
And what happens next to the DNA process?
So the next step will be those tubes that just went through the heating and cooling, that those tubes will be used for the 4th step of separation.
And who did that step?
Troy Adams.
So you did not do that step?
No, I did not.
And did that end your work on this case?
Yes, it did.
Any cross-examination? You may proceed.
Thank you. Can you state your name for the court, spelling your first and last name?
My name is Troy Adams, Troyadams.
Thank you. And what is your educational background, sir?
I have a bachelor's degree from Pennsylvania State University in forensic science.
And what is your current occupation?
So I currently work as an investigator for the Lehigh County Coroner's Office in Pennsylvania.
And how long have you been in Pennsylvania?
I've been in Pennsylvania for approximately 3 years now, 2 years working in my current job.
So prior to moving to Pennsylvania, did you work in a different field?
No.
Did you work for forensic sciences in Massachusetts?
Yes.
And can you detail your working history as it pertains to forensic sciences?
Yes. So out of college, I joined the Massachusetts State Police crime lab there as a DNA analyst. I completed all my training protocols and was tested for competency, and then I began performing casework on samples.
And then, did you work for a different company in Pennsylvania as well, doing forensic sciences?
Yes. After I left the Massachusetts State Police, I joined Quest Diagnostics as a forensic scientist in their toxicology division.
And just keep your voice up so everyone can hear you.
And have you had training for your position at the Massachusetts State Police Crime Lab and also for your position at Quest?
Yes.
And have you had ongoing training as well?
Yes.
And have you taken proficiency tests?
Yes.
And what were the results of your proficiency tests?
I passed every proficiency test.
And do you have any certifications in your current job?
Yes.
And where are those from?
So I am certified as a national medical legal death investigator.
And how did you receive that?
So I received that after a certain amount of training at my current job, and then going through and taking a certification test.
I want to direct you now to when you were working in Massachusetts. Did you do work on lab number 23-00596?
May I refer to my file?
You may.
Yes.
And did you do work on items 9-27.2.1, a sample from interior of a dark gray slipper, 9-27.3.1, stain A on the exterior left gray slipper, 9-32.1.1, the sample from the interior sleeve cup of a Tyvek suit, 9-32.2.1, the sample from stain A on the exterior left pant leg of Tyvek suit, 9-32.5.1, the sample from stain D on the interior right sleeve of the Tyvek suit, and 16-13.1, the unknown tissue recovered from the Swampscott dumpster.
Yes, I did.
And what work did you do on those 6 samples?
So I performed the second and fourth step of the DNA analysis process, quantification and detection.
And can you tell us how you did the quantification step?
So I took a sample, I was given samples from the extraction step, and then I added a mix of chemicals and went through repeated processes of heating and cooling, which quantified the DNA.
And then the process was then next ready for the third step, is that correct?
Yes, it was ready for amplification.
And who did the amplification step?
I believe that was Madison Frank.
Okay. And after Madison Frank did the amplification step, what did you next do?
I took the products of the amplification step and performed detection.
And did you do the detection on those same 6 items that we just discussed?
Yes.
Okay. And how did you do the fourth step, that detection?
I added a mix of chemicals and placed the samples on the detection analyzer.
And then what happened?
And then electropherograms were generated, which were then interpreted by the interpreting analyst.
And where did that data go?
That data was transferred to one of our servers at the State Police Crime Lab.
And did you do the interpreting or did someone else do it?
Someone else performed the interpretation.
Did you do any other work on this case?
Not that I'm aware of, no.
Okay. I have no further questions.
Any cross-examination of this witness? The witness may step down.
This way. Just watch your step.
You may proceed.
Thank you. Good afternoon. Could you please state your name for the court?
My name is Emily Oliver.
And can you tell us your educational background?
Yep. I received a Bachelor's of Science in Forensic Science from the University of New Haven in 2019.
And what is your occupation, ma'am?
I am a forensic scientist 2 at the Massachusetts State Police Crime Lab in the DNA unit
And how long have you been employed there?
since December 2019.
And can you please tell us your responsibilities?
So at the lab, I perform DNA testing on samples as well as DNA profile interpretation, and I write lab reports.
And what training have you had for this position?
So when I was first hired, I did an in-house training program that lasted over 6 months that involved testing mock casework samples as well as lectures and exams.
And you also have ongoing training.
Yes.
And have you taken competency tests and proficiency tests as well?
Yes.
And have you passed those?
Yes.
Prior to doing any work, how do you prepare your work area?
So first I clean the area and all the tools I'm going to use with bleach, and then I lay down clean bench paper.
And how are you dressed before you work?
I have a lab coat, gloves, a face mask, and a hairnet.
And why do you take those precautions?
To prevent any contamination from myself onto the samples.
I want to direct your attention now to lab case number 23-00596 and asked if you worked on items 7-13.1, sample from area A on the head of the hatchet, item 7-13.2.1, sample from the handle of the hatchet, and item 9-20.1, the sample from stain A on tape with apparent gauze with red-brown stain. Did you do work on those items?
I did.
And what did you do?
So for 7-13.1, the head of the hatchet sample, I did the first 3 steps of our 4-step DNA analysis process, which is extraction, quantification, and amplification. And on the other 2 samples, I did the first 2 steps of our 4-step process in the lab.
Okay, I also want to ask you if you did any sample prep.
So I did, and the sample, it was more so just making sure I pulled the correct tube with the correct case number and item number from our cold storage room. It was previously prepared by someone else.
Thank you. Can you tell us what you did for the first step for the items that you did the first step for?
So extraction is the step where we add chemicals and heat to the sample to release the DNA from inside of the cells in the sample.
Okay. And which items did you do that for?
For 7-13.1, 7-13.2.1, and I believe it was 9-20.1.
Okay. So for all 3 of those, you did
Yes.
that. Okay. And did you use equipment for the extraction step?
Yes.
And what equipment did you use?
It's called an Easy One extraction robot.
And what is the biological material now referred to?
A DNA extract.
And what is it now ready for?
The second step, which is quantification.
And how did you perform the quantification?
So I took a small droplet from the extract tubes and put them into a new tube. And then I add different chemicals to that new tube and put it through an instrument that estimates the amount of DNA in the sample.
And was that for all 3 of those items?
Yes.
Okay. And did you use equipment?
Yes. It's called a 7500 real-time PCR instrument.
Okay. And then what happened in the DNA process?
And after that, the samples go to the third step if they have enough DNA, which is amplification.
Okay. And for which items did you do the amplification process? Did you just do that for 7-13.1?
Yes.
Okay. Why did you not do amplification for 9-20.1 and 7-13.2.1?
Just due to scheduling reasons, that was worked best for me.
Okay. So who performed the amplification process for 9-20.1 and 7-13.2.1?
My coworker, Mary Nagel.
Okay. So talking now about what you did do amplification for, item 7-13.1, the sample from area A on the head of the hatchet. Can you tell the jury how did you perform the amplification process on that item?
Sure. So I add, again, different chemicals to the extract tubes and put it onto an instrument that creates millions of copies of these specific locations on the DNA that we're looking at.
And did you use equipment?
Yes, it's called a thermocycler.
And what happens next in the DNA process?
The last step is the separation and detection step. And that after that last step, we have the DNA profile.
And did you do that fourth step, the detection step?
I did not.
And who did that last step for 7-13.1?
My coworker, Yoli Bregu.
I have no further questions, Your Honor.
I just have a few.
Go right ahead.
And first off, Oliver. I didn't hear your name correctly.
Oh, Emily Oliver.
And do you have a title? Kristen?
You can just call me Miss Oliver if you'd like.
Miss Oliver. Can you tell this jury a little bit something about your knowledge of inhibitors that might affect the amplification of the work?
Sure. So different materials in the environment can kind of limit the reaction and which could affect the DNA profile that we get in the end.
And can you just tell the jurors what are some of the known inhibitors of DNA amplification?
So dye from blue genes can be an inhibitor. I don't recall any others off the top of my head.
All right. The fact of the matter is that the DNA molecule is a pretty robust molecule, correct?
Correct.
And it's fair to say that things as common as oil and things like grease, they don't inhibit the amplification of the DNA molecule in this testing, do they?
I'm not sure.
What about the quantities that you're dealing with? Is it fair to say that the quantity that you're amplifying can be measured in nanograms?
Yes.
And what's a nanogram?
I believe it is, so it's just a very small number. I believe it's 1 × 10⁻12.
So is it fair to say that it's about a one billionth of a gram?
I'm not sure.
Okay.
But,
And what about a picogram? Can you define a picogram for the jurors?
it is a slightly larger number, but still a very small quantity.
Okay. And in the amplification process that you just told the jurors about, what happens is that you separate the alleles from the molecule, the DNA molecule, and then you amplify those alleles, and then they reunite into a molecule, and a DNA molecule, and then your machine and apparatus that you use, it's able to process it and give a result, correct?
Correct. And that's mostly referring to the fact that DNA is a double helix, kind of looks like a ladder that's twisted, and it becomes single-stranded during part of the process, and then comes back together again.
All right. And finally, you're familiar with the term electropharogram, correct?
Yes.
And can you just describe for the jury what an electropharogram is?
So it's just a graphical representation of the DNA profile, kind of like an EKG. There's a bunch of different peaks that correspond to the DNA. That's a printout that we get and that we look at the end of our testing.
So in the State Police Crime Lab, when you look at the electropharogram, does the analyst interpret the peaks to determine the results of the DNA testing, or is it another piece of apparatus that does the analysis of the electropharogram peaks?
So we put our electropharogram through a program called Gene Mapper that does help the interpretation, but a person is still looking over the profile.
All right. So is it fair to say that in the State Police Crime Lab that the final call, so to speak, the final determination of what is or isn't interpreted as DNA, that's done by the analyst, not by the machine?
Correct.
Thank you, Ms. Oliver.
Okay, thank you.
Any redirect? The witness may step down. You may proceed.
Thank you very much, Your Honor. Can you state your name for the record and just spell your last name?
Yep. My name is Mary Nagel, Nagle.
And what's your educational background, ma'am?
I have a bachelor's of science in biochemistry, cell and molecular biology from Drake University, as well as a master of science in biomedical forensic sciences from Boston University.
Can you just pull that microphone a little bit closer? Thank you. And what is your occupation?
I am a forensic scientist too in the DNA unit at the Massachusetts State Police Crime Lab.
And how long have you been doing that?
Just over 3 years.
Can you tell us your responsibilities?
Yep, I am capable of performing all of the lab work as well as reporting and interpreting single source profiles and writing up other reports as well.
What training have you had for this position?
I underwent a 6-month training program that included lectures, exams, and about 50 mock samples for lab practice.
And did you pass the exams that you during your training period?
Yes, I did.
And do you also have ongoing training?
Yes, I do.
And as part of your ongoing training, do you have to take proficiency tests?
Yes, we take 2 proficiency tests a year.
And have you passed those tests?
Yes, I have.
Directing your attention to lab case number 23-00596, did you do work on item 7-13.2.1, the sample from the handle of the hatchet, and item 9-20.1, the sample from stain A on tape with apparent gauze with red-brown stains?
Yes, I did.
And what did you do on those items?
I performed the 3rd and 4th step of the DNA analysis, which is the amplification and detection process.
And how did you do the amplification step?
For amplification, I was made aware of which samples to process from Emily Oliver. From there, the samples were set aside in a tube, and I took whatever amount was needed specific to that sample item and added chemicals to that. The samples and the chemicals were then put on an instrument that went through a series of heating and cooling, and this process makes millions of copies of that DNA.
And did you use equipment?
Yes, for that, it is mostly just the heating and cooling instrument that is used, and then the pipettes and tubes and instruments like that as well.
And then you said you did the 4-step detection?
Yes, I did.
And which did you do the detection for both of those items that we previously discussed?
Yes, I did.
Okay. And how did you do that?
So after the 3rd step amplification, the samples are in smaller tubes, and a small portion of that sample is then loaded onto a plate with another set of chemicals. That plate is loaded into an instrument to run, and that instrument then detects the DNA and produces a profile for interpretation.
And did you do the interpretation?
No, I did not.
Who did the interpretation?
That was Salmon Salem.
I don't have any further questions, Your Honor.
Any questions of this witness? You may step down, thank you.
Thank you.
Oh, I'm sorry.
I need to speak up. Ms. Nagel?
Yes.
Just have a few questions for you. Once again, there are certain known inhibitors that affect the ability to amplify the DNA molecule, correct?
There are, yes.
But there's not very many,
Correct.
because it's a robust molecule, correct?
Right.
One of the inhibitors is, interestingly enough, the blue dye that we see in things like Levi's, correct?
It can be, yes.
Okay. But it's not something like oil or grease or things like that. The molecule doesn't get broken down by oil and grease and things like that, correct?
Correct.
And If bleach is used on an item that contains DNA and the bleach breaks down the molecule, that molecule will not properly amplify using your equipment.
It will still be amplified, but parts of that DNA will probably drop out at the end of it, but there is still usually DNA at the front of that profile.
When you say at the front, you mean that the shorter alleles, after the molecule is broken down, the shorter alleles will amplify, but the longer alleles won't because they were broken down by the bleach.
Correct.
And when you look at the results of their amplification, a trained analyst can see that the longer alleles didn't amplify properly, and they may conclude that something like that particular sample might have been affected by bleach or something like that, correct?
We do not look into what could possibly affect the degradation, but we are capable of seeing the fact that the DNA profile will begin to slowly drop in what is present.
Okay. Thank you. One other question. Am I correct in saying that a nanogram is 1/billionth of a gram?
Yes, I believe so.
Okay. Am I correct in saying that a picogram is 1,000th of a nanogram?
A picogram would be 1/millionth.
And when you're using the quantities that you're using to get the results that you complete in the lab, we are talking about quantities in nanograms, aren't we?
Yes.
Tiny, tiny amounts, correct?
Yes.
Thank you. Thank you very much.
Any redirect of this witness?
No, Your Honor.
The witness may step down. You may proceed.
Thank you, Your Honor. Good afternoon.
Good afternoon.
Could you please state your name and spell your entire name for the record?
My name is Yoli Bregu, Jolibregu.
And what is your educational background?
I have a Bachelor of Science in Biology from Boston University and a Master of Science in Biomedical Forensic Sciences, also from Boston University.
And what is your occupation, ma'am?
I'm a forensic scientist too with the Massachusetts State Police Crime Laboratory in the DNA unit.
And how long have you been there?
Since June 2019.
Can you tell us your responsibilities as a forensic scientist too?
Sure. I perform forensic DNA analysis on evidence samples submitted to the lab. So my typical workday consists on processing those samples in the lab, doing the data interpretation, making comparisons with the reference samples, writing reports, testifying in court as needed. I also do administrative and technical reviews.
And what training have you had for this position?
So in addition to my education during my master's degree, I followed a forensic biology and forensic DNA analysis track. And then when I got hired, I was trained at the Massachusetts State Police, specifically in the DNA unit in forensic DNA analysis. The training consisted on lectures, reading of relevant literature, laboratory experience with practice samples, data interpretation, written and oral exams, followed by competency tests.
And did you pass those competency tests?
Yes.
And do you have ongoing training as well?
Yes, we have ongoing training. Every year we are required to take at least 8 hours of continuing education, which is offered in forms of seminars, webinar, workshops. We usually take more because there's more offered through the year. We are also required to read at least one scientific journal article relevant to our field.
And you have to take testing as well?
Yes, we have to take proficiency testing every year. They are taken twice a year.
And have you passed that proficiency testing?
Yes, I have passed the proficiency testing required throughout my employment. The last one is still in the grading process.
And have you testified in court about DNA?
Yes, I have.
Directing your attention to lab number 23-00596, did you do work on item 7-13.1, the sample from area A on the head of the hatchet?
Yes, I did.
And what work did you do on this item?
I performed the fourth step that is used in forensic DNA analysis, which is detection.
And how did you do the detection step of the DNA process?
So I took the samples from the analyst who had done previous work, Emily Oliver. I prepared those samples, mixing them with the required reagents. Then I loaded them in our instrument that does the detection, known as genetic analyzer, and I started the run. And that's where my role ended. After the run is done, the data is saved automatically in the computer and can be imported by the interpreting analyst for data interpretation.
Did you do the interpretation here?
No, I did not.
Who did the interpretation?
Salmon Salem.
I have no further questions, Your Honor.
Any cross-examination of this witness?
Just a couple.
Go right ahead.
Thank you. Mr. Booth,
Yes.
did I see that right?
Yes, you did.
A couple of questions. You're familiar with the term in the context of DNA testing, allele, correct?
Correct, yes.
And the allele is, why don't you just tell the jury what?
The allele is a variant of a DNA fragment that is amplified during our third step. So it's a person has two alleles, one they inherit from the mother and one from the father.
Okay, and the allele then is the section of the DNA molecule that is then looked at once you've separated the molecule, and it's the part of the molecule that then gets amplified, correct?
Yes.
And so if you have an allele that repeats at a certain locus on the DNA molecule, then you may be a particular type at that particular location on the molecule, depending on how many repeating alleles you have.
It's how many repeats are within the allele. So the allele itself is the entire fragment, and within that alleles, there are we call them short tandem repeats. There are repeats, they repeat themselves different numbers for different individuals, and that's what it's assigned to an allele. How many times the fragment repeats itself within the allele.
And people can have the same repeats of alleles in the molecule, but generally speaking, they don't have the same number of repeating alleles across the molecule, various different loci.
Yes, correct.
And the loci is just plural for locus, right?
Yes, the loci is the location of that particular allele in the DNA molecule.
In the testing that was done in this case, you're using 26 different locations plus the sex area.
Correct, yes.
And that's a nillion.
Correct, yes.
Thank you.
Any redirect of this witness? No redirect. The witness may step down.
Thank you, Your Honor.
Thank you. You, the judge. Days ago, I told you, and I think I read you another stipulation, but I'll remind you, a stipulation is just an agreement between the parties, and I'm going to read you now an agreement that the parties have made. This is a stipulation regarding DNA samples of Anna Walshe and Brian Walshe. It reads as follows. On May 9, 2018, FBI Special Agent Tyler Delorme obtained a DNA sample from Brian Walshe. Special Agent Delorme brought the sample to the FBI headquarters in Boston, where it was placed into evidence storage. Also on May the 9th, 2018, FBI Special Agent Matthew Riportella obtained a DNA sample from Anna Walshe. Special Agent Riportella brought the sample to the FBI headquarters in Boston, where it was also placed into evidence storage. On January 10th, 2023, Special Agent Kristen Koch took the sample of Anna Walshe and the sample of Brian Walshe from evidence storage at the FBI headquarters in Boston and brought both samples to the Massachusetts State Police Crime Lab. The samples did not leave the custody of the FBI until Special Agent Koch submitted them to the Massachusetts State Police Crime Lab for testing. That's Exhibit E for identification. Commonwealth may call its next witness.
The Commonwealth would call Karen Jacobson.
Do you solemnly swear or affirm the testimony you'll give in the cause now in hearing should be the truth, the whole truth, and nothing but the truth, so help you God? Thank you.
Right this way. Just step for me. Further.
Thank you.
You may proceed.
Thank you very much, Your Honor. Good afternoon.
Good afternoon.
Could you please give us your name and spell your name for the record?
My name is Karen Jacobson. That's KarinJacobsen.
And if I could just ask you to pull that microphone a little closer to you. Thank you.
Sure.
What is your occupation, ma'am?
I'm currently employed as a forensic scientist 2 within the DNA unit at the Massachusetts State Police Crime Laboratory.
And how long have you been there?
Since August of 2021.
And what are your duties?
I'm responsible for receiving items of evidence associated with known individuals, taking them through the 4-step DNA process, and making those DNA profiles available for use in comparisons.
And what is your educational background?
I have both a Bachelor's of Science in Forensic Science and a Master's of Science in Forensic Science from Bowling Green State University.
And do you have specialized training to be a DNA analyst?
Yes, I do.
And can you tell us about that training?
The training program within the DNA unit is at least 6 months long. It consists of lectures covering all of our protocols, literature readings covering current and historic methodologies, laboratory processing of at least 50 mock samples, and all of this is wrapped up with a competency test.
And did you pass your competency test?
Yes, I did.
And have you also had ongoing training?
Yes, I have.
And as part of your ongoing training, do you have proficiency testing?
Yes, I do.
And have you passed your proficiency training?
Yes, I have.
I want to ask you about lab number 23-00596. Did you do work on this case?
Yes, I did.
And what work did you do on this case?
The DNA testing process is 4 steps long. I performed the 2nd step and the 4th step for 2 items.
And what were those items?
These were items 13-1.1 and 13-2.1.
And what were those?
These were the DNA saliva collection kits for Anna Walshe and Brian Walshe, respectively.
Okay. So how did you do the step 2? That be the quantification step?
Yes.
Okay. Tell us about that, please.
In the 2nd step, quantification, I received a portion of the extract created in the 1st step. I applied a set of chemicals to it and put it on an instrument that goes through cycles of heating, cooling, and this produces an estimate of the amount of human and male DNA. Walshe and Brian Walshe, respectively.
Okay. So how did you do the step 2? That be the quantification step?
Yes.
Okay. Tell us about that, please.
In the second step, quantification, I received a portion of the extract created in the first step. I applied a set of chemicals to it and put it on an instrument that goes through cycles of heating, cooling, and this produces an estimate of the amount of human and male DNA present within each sample.
And did you use equipment?
Yes, I did.
Okay. And what was that?
This is a 7500 real-time PCR machine.
And who had done the previous step?
The previous step was performed by Katarina Stashin.
Okay. And why didn't you do that step?
We batch our samples in the laboratory processing for the sake of efficiency.
Okay. And then when did you become involved again in this process for these samples?
I then performed the fourth step, separation and detection.
Okay. And can you please tell this jury how you did that fourth step?
So the same person, Katarina Stashin, performed the third step. I received the product produced in the third step from her. I applied another set of chemicals to it and put it on an instrument that separates each of the copies of the DNA by size. And then with the help of some special software, it turns it into a visual representation of the DNA profile.
And did you use any equipment?
Yes, I did.
And what equipment did you use?
This was a 3500 genetic analyzer.
Okay. And then what is the data next ready for in the process?
After the data is reviewed, it's ready for use in comparisons.
Okay. And who did the interpretation and comparison here?
In this case, it was Katarina Stashin as well.
And did you do any other work on these samples?
No, I did not.
I have no further questions.
Cross-exam? The witness may step down.
Thank you.
Oh, just step forward.
Thank
Step
you.
forward. A little bit further. There you go.
You may proceed.
Thank you very much, Your Honor. Good afternoon. Can you please state your name and spell your whole name for the record?
Good afternoon. My name is Katarina Stashin. First name is spelled Katarina. Last name is spelled Stashyn.
And what is your current occupation, ma'am?
I'm currently a criminalist in the biology section at the Washoe County Sheriff's Office Crime Lab located in Reno, Nevada.
And can you please detail your work history as it applies to forensic sciences?
I was previously a forensic scientist in the DNA unit at the Massachusetts State Police Crime Laboratory for about 3.5 years, from 2021 to earlier this year, 2025, when I got the opportunity to move back to Nevada to be closer to family and pursue a job still as a DNA analyst.
And what is your educational background?
I have a bachelor's and master's degree in biotechnology from the University of Nevada, Reno.
And do you have any specialized training to be a DNA analyst?
Yes, I received training at both the Massachusetts State Police Crime Lab and at my current job in Washoe County. In Massachusetts, I completed an approximately 1-year-long training program that consisted of reading scientific protocols, scientific literature, textbooks, all on the topics of forensic DNA analysis. I also took written exams and oral exams on those topics. I also practiced sets in the lab, both for laboratory processing and for DNA interpretation. Put together practice case files, wrote reports, did comparisons, and all of that training completed with a competency test, of which I passed. And then at Washoe County, I completed an approximately 6-month-long training course due to my experience, where I also did review of scientific literature, textbooks, protocols, practice sets, writing practice reports, performing comparisons or interpretation, et cetera. And that also completed with a competency test, of which I passed.
And have you also had proficiency testing?
Yes, I have also completed proficiency testing.
And have you passed those tests as well?
Yes, I have passed all competency and proficiency testing that I've taken so far.
And have you testified in Massachusetts about DNA?
Yes, I have.
So what is a single source DNA profile?
A single source DNA profile is a profile that originates from one person or one contributor. And you can identify this in a profile. Just by looking at the profile, you can see how many people are in there.
And what is a known standard?
A known standard is a sample that's taken directly from an individual. For example, a sample from somebody's mouth, a saliva swab, or a blood sample. It's taken directly from a person to be used for comparison purposes.
And how is a known standard developed into a DNA profile?
A known standard will go through the four-step laboratory process to result in a DNA profile that can then be used for comparisons.
And the four-step process is what?
The four-step laboratory process for DNA analysis is extraction, quantification, amplification, and detection.
So prior to doing any work, how do you prepare your work area?
So when you first get into the lab to do any of those steps, you're going to clean your lab bench. So that involves using a bleach spray to wipe down your area. You're going to clean all of the equipment, utensils, your pens, pipettes, any equipment that you're using. And then me as a person or any lab analyst, you'll be wearing a lab coat, a face mask, a hairnet. You're in full PPE, wearing gloves, changing gloves frequently.
Why do you clean your area and wear PPE?
We do this just to keep everything perfectly clean, or the cleanest that we can, just to help the integrity of the evidence and help produce the best possible profile.
Now I'm going to direct your attention to Massachusetts Case Lab 23-00596. Did you do work on this case?
Yes, I did.
And what did you do on the known standards in this case?
I performed the sample prep, the extraction, and the amplification on the known standards in this case.
And did you also do interpretation on this case?
Yes, I also reviewed the profiles that were obtained from that process and made sure they were able to be used for comparison purposes.
So I want to ask you about each of the things that you just told us about. So were you working on 13-1 and 13-2, is that correct?
Correct.
And what were those?
Those were the known saliva standards for both Anna Walshe and Brian Walshe.
And so what was 13-1?
13-1 was the known saliva standard for Anna Walshe.
And what was 13-2?
13-2 was the known saliva standard for Brian Walshe.
And how did you do the sample prep for 13-1?
So both of these samples were known saliva samples, so a swab from the mouth, and I just cut up the swab and placed the cotton part of the swab into a tube, a labeled tube with those item numbers that I said.
Okay, so that's what you did for 13-1, and then did you give it another number?
So once I cut that swab off, it then became 13-1.1.
And so did you do the same thing for 13-2?
Yes. So then the cotton that got cut off of the whole swab became 13-2.1.
And then after you did that sample prep, what did you next do?
Then those samples went through the extraction step of the process.
And how did you do the extraction step?
So extraction consists of adding chemicals to the samples and going through heat variation to help break open the cells present in the sample and release the DNA that is present, and then that DNA is isolated.
And did you use equipment for that?
Yes.
What equipment?
An incubator was used for that heat variation that I mentioned, and an Easy One robot was used for the extraction part, meaning that after it sat in the incubator with the chemicals to break open the cells, a robot is used to isolate that DNA from the rest of the cellular debris left behind.
So you told us about sample prep and step 1. Did you do the next step, step 2?
No.
Why not?
It's just based on scheduling at the lab, who was assigned to do what step that day.
And who did that next step?
The second step of quantification was performed by Karen Jacobson.
Were you qualified to do all of the steps?
Yes.
And so what did you next do?
I next did the amplification step, that third step I mentioned.
And how did you perform the amplification step?
So amplification is the third step, and this is when we make millions and millions of copies of certain fragments of DNA. So this is done by, again, adding certain chemicals and going through heat cycles to make these many millions of copies of DNA. Because we're usually working with a smaller amount of DNA, you need to make a lot of copies to make it visual and able to be detected.
Did you use equipment?
Yes.
What equipment?
We use what is called a thermal cycler, and this is the instrument that performs those heat cycles needed to make the copies.
So did you do the next step, the fourth step?
No.
Who did that?
Karen Jacobson also performed the fourth step of detection.
So then what did you do next?
So after that full laboratory process was completed, I looked at the data that was produced from those 4 steps, and both items resulted with DNA profiles that I reviewed and made sure were acceptable to be used for DNA comparisons.
And could you visualize a single source?
Yes.
Right. And did you generate a DNA profile for Anna Walshe?
Yes.
And did you generate a DNA profile for Brian Walshe?
Yes.
And were profile sources developed that were appropriate for comparison?
Yes, the profiles developed were acceptable to be used for comparisons.
And they were both single sources?
Yes.
What does appropriate for comparison mean?
When I'm saying appropriate for comparison, I mean that the profile is full, as in data is obtained at every location that was tested, and that it is single source.
And what do you do with the profiles?
That profile is then made into a text file that is made available for the analyst that's going to be doing the comparisons.
And were the profiles assigned a number to identify them?
Yes. So the numbers for the profiles are the same as their item number. So the text file profile for Anna Walshe also had the number of 13-1.1, and the text file profile for Brian Walshe was 13-2.1.
If I might have 1 second, Your Honor.
You may.
Nothing further, Your Honor.
Cross-exam. The witness may step down. So help you God.
Yes, I do.
Thank you. You can be seated.
Right this way. Just watch your step.
You may proceed.
Thank you. Good afternoon.
Good afternoon.
Could you please introduce yourself to the jury by stating your full name and spelling your name?
My name is Salmon Salem, S-A-M-A-N. Last name is S-A-L-E-E-M.
And could you please tell us your educational background?
I have a bachelor's degree from Boston College in biochemistry and a master's of science in forensic science from Boston University.
And what is your occupation, ma'am?
I currently work as a forensic scientist 3 at the Massachusetts State Police Crime Lab within the DNA unit.
And what is a forensic scientist 3?
A forensic scientist 3 is a supervisory position within the DNA unit where I manage other analysts, forensic scientist 1 and 2, that work within the DNA unit.
So how many people do you supervise?
I supervise 6 people currently.
And what are your responsibilities in supervising those people?
As a supervisor, I manage their caseload by assigning them cases to work on in the laboratory, as well as for interpretation of DNA profiles for evidence items that are submitted to the DNA unit. I also train them in the methodologies for DNA testing. For example, if there's any new hires in our laboratory, I would be part of their training program where I would teach them about the laboratory work that's conducted, as well as give them presentations on the theoretical background of DNA testing. In addition to that, I also work on my own cases, so where I'm assigned cases by my manager, and I do the interpretation of DNA testing on those cases and review the cases of other people and what they've worked on.
And how long have you been employed at the Massachusetts State Police Crime Lab?
I've been there approximately 10 years.
Do you have any specialized training to be a DNA analyst?
When I first began at the DNA unit 10 years ago, I went through an 8-month-long training program where I learned about the theoretical discipline of forensic DNA testing, as well as the practical application of it. I tested mock evidence samples in the laboratory to determine what those DNA profiles were and learned about how to do that testing. Additionally, since that training, every year we go through continuing education where we learn about new methodologies that we're adopting in the laboratory and also any kind of changes that are happening in the field of forensic DNA analysis.
So you said that you've had testing, is that correct?
I'm sorry.
Have you had testing?
Yes.
And what kind of testing have you had?
So every time there's a new technology adopted or there's new training that I go through, at the end of that training, we do a competency test where we are tested on if we have understood that methodology by taking a written exam or oral exam as well as a practical exam in the laboratory. In addition to that, twice a year, we go through proficiency testing.
So have you passed the competency testing that you've taken?
Yes, I have.
And you said that twice a year you have proficiency testing. Who monitors that or provides that proficiency testing?
So the proficiency test samples are provided by an external agency, and those samples are tested in our laboratory as we would process any casework samples as well. And then the answers for those items of evidence, the DNA profiles we obtained, are submitted back to that external agency who will then grade us to ensure that we obtain the correct DNA profiles.
And have you passed that proficiency testing?
Yes, I've passed all the ones I've taken over the 10 years.
Thank you. Are you familiar with lab case 23-00596?
Yes, I am.
And what type of DNA testing was conducted in this case?
In this case, we conducted a type of testing known as STR DNA testing, which follows a 4-step process in the laboratory, at the end of which we obtain a DNA profile.
Okay. So just generally, can you discuss that 4-step process?
Generally, that 4-step process starts with extraction in the first step, where we break open the cells in which the DNA is found. This is similar to like cracking open an egg, where the DNA is found within the yolk of it, and we extract that DNA. We use heat and chemicals to do this. The second step is known as quantitation, where we determine how much DNA is present in that sample so that we can proceed with the next step. The next step is known as amplification. In that third step, we make millions and millions of copies of the DNA that is found there, targeting certain locations within the DNA. In this case, we target 26 locations that are found throughout the chromosome, as well as a sex determining locus as well. And then the last step is capillary electrophoresis. During that step, we analyze those copies of DNA to generate a DNA profile, which can then be interpreted for comparison purposes.
And then once the raw data is generated, you said that you're able to make some comparison here?
Yes.
And how is that done generally?
So generally, the first thing we would do is interpret the DNA profile, which means that we would observe that DNA profile to see what is in that DNA profile. So what are the different characteristics of the DNA that we're observing at these 26 locations that we have tested. We will determine how many individuals are in this mixture, so the number of people that make up this DNA profile. Is it coming from one person or multiple individuals? And then that profile, once we've determined what it is, we can compare it to a known standard or somebody whose DNA profile we know to see if they would be included in that sample or excluded from it.
And in this case, 23-00596, there were many items that were tested by multiple people in the lab for DNA testing. Is that correct?
That's correct.
And all of those people did the 4-step process. Is that correct?
That's correct.
And what was your role here?
At the end of that 4-step process, data is generated on a computer. At the end of that process, my role comes in at that point where I look at that data. So it comes off in a form that we have to interpret using a software program. So that's where my role starts, where I interpret that data.
So I'm now going to start to ask you about what you did here and about each individual piece of data that you looked at with each individual item that you looked at. Is that okay?
That's okay.
So let's start with the first round of testing and the first items that you looked at. I want to ask you about item 9-27.2.1. What is that item, ma'am?
Do you mind if I look at my notes?
You may look at your notes.
Thank you. That is a sample of an interior of dark gray slippers found in bag number 4 at 300 Forest Street.
And what was the DNA profile result for this item?
This DNA profile was interpreted as a mixture of 3 contributors, including male DNA.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
Yes. The DNA profile from this item is at least 270 million times more likely if it originated from Anna Walshe and 2 unknown individuals than if it originated from 3 unknown unrelated individuals. This provides support for an inclusion of Anna Walshe to this profile.
So what does that mean?
So the statistic we report is known as a likelihood ratio. The first part of this is determining what the DNA profile is. In this case, the DNA profile is a mixture of 3 contributors, meaning that the profile that was observed on this item came from 3 individuals. In this profile, when we do a comparison, we compare the characteristics at each of these 26 locations to see if the profile that is Anna Walshe's profile matches those or is consistent with the ones that are observed in the DNA profile. And through the statistical analysis, what we learned is that it is at least 270 million times more likely to observe this DNA profile if it originated from Anna Walshe than if it did not, and 2 other individuals than if it originated from 3 unknown unrelated individuals. The likelihood ratio is a statistical calculation that shows us how likely it is to observe a DNA profile given two propositions. One, that the person would be included in the DNA profile, or the other, that the person is excluded or not included in the DNA profile. And the value of that likelihood ratio can tell us which of these propositions is more likely to observe this DNA profile.
So how do you calculate the likelihood ratio?
That is calculated using a software known as STR-Mix or STAR-Mix. And in that software, we use calculations that we derive from the general population of how common or rare certain characteristics are at these 26 locations observed. And we use that in the general population in the US, and we use that information to calculate the likelihood ratio.
Can you explain when you said this was a mixture of 3 contributors?
So that just means that the DNA profile that we observed at these 26 locations, what I observed is that I would see DNA from multiple people, in this case, 3 individuals. We receive half our DNA from our mother and half from our father. So we would expect to see for a single source profile or something coming from one person would either have one characteristic or 2 characteristics at each of these locations. In some of these locations, I saw 5 or 6 characteristics, which means that there's at least 3 people here in this mixture.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Yes. The DNA profile from this item is at least 12 quintillion times more likely if it originated from Brian Walshe and 2 unknown individuals than if it originated from 3 unknown unrelated individuals. And this provides support for an inclusion.
What is a quintillion?
Quintillion is 1 followed by 18 zeros.
Can you tell when the DNA was contributed?
I cannot, through the testing that we do, we cannot determine when or how the DNA was deposited on an evidence item.
Does DNA have to be contributed at the same time?
No, it does not.
Moving to item 9-27.3.1, can you tell me what that item is?
That was a sample from stain A on the exterior top left of the gray slipper, bag number 4 at 300 Forest Street.
And can you tell me what's the DNA profile result for this item?
This DNA profile was interpreted as a mixture of two contributors, including male DNA.
Were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
Yes. The DNA profile from this item is at least 16 nonillion times more likely if it originated from Anna Walshe and an unknown individual than if it originated from two unknown unrelated individuals, and this provides support for an inclusion of Anna Walshe to this profile.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
The DNA profile from this item is at least 77 times more likely if it originated from Brian Walshe and an unknown individual than if it originated from two unknown individuals, unknown unrelated individuals. This provides limited support for an inclusion. Our DNA unit validation studies have shown that there is an inclusion of non-contributors in this range, likelihood ratio range of about 2 to 1,000. And adventitious support could be attributed to this conclusion.
So what does that mean?
So when we do these calculations for the likelihood ratio, we're relying on the DNA profile that we observed in that evidence item. The more data we have there, that means the more of these 26 locations where we observed this DNA profile, the higher the statistic will be for an inclusion. And that's because there's more support and more data observed. When there is limited support for an inclusion, it means that the data for that contributor in that mixture, so it doesn't mean everyone in that mixture had less DNA, just that one contributor, one or more contributors had less DNA available there for comparison, so the statistic is lower because of it. In our validation studies, what we did see, because all humans do share some DNA with each other, that we would see some non-contributors, meaning that people who really shouldn't we made mixtures where we knew who was in those mixtures. And so we would have some non-contributors get a likelihood ratio that's inclusionary, show that they're included when they really shouldn't be included. And it's because they happen to have some alleles that are common with what was observed in the DNA profile. So in this case, for the statistic, even though there is support for an inclusion, that support is limited, meaning that there is less data available.
What is a nonillion?
A nonillion is 1 followed by 30 zeros.
What type of cells is DNA found in?
It is found in all kinds of biological cells that we have, such as our blood cells, which are our white blood cells, our hair cells, skin cells, saliva, anything we could have in our body that has a nucleus. Any cell that has a nucleus would have DNA in it.
And what of those is potentially found in a shoe?
From a shoe, it could be if there's a biological process that's deposited on there, that biological fluid could contribute DNA, or more typically for clothing and shoes, it would be from skin cells that are from wearing that shoe multiple times.
Do your results tell you what type of cells these came from?
No, they do not.
And do the individuals have to contribute the same type of cells?
No, they do not. One of the things that could cause a mixture of DNA profiles is if there's different types of cells from different individuals that are contributing to the same evidence item.
What is item 9-32.1.1?
That is a sample of an interior sleeve cuff of a Tyvek suit that was found in bag number 6 at 300 Forest Street.
And what was the DNA profile result for this item?
The DNA profile for this item was interpreted as a mixture of 3 contributors, including male DNA.
Were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
The DNA profile from this item is at least 4.8 nonillion times more likely if it originated from Anna Walshe and 2 unknown individuals than if it originated from 3 unknown unrelated individuals, and this provides support for an inclusion.
And were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Yes, the DNA profile from this item is at least 100 trillion times more likely if it originated from Brian Walshe and 2 unknown individuals, than if it originated from 3 unknown unrelated individuals, and this provides support for an inclusion.
So when you say things like this supply provides support for an inclusion, is that standard language that you're using?
Yes.
And standard language for whom?
The standard language for forensic DNA analysis in general, and then at our laboratory, we've adopted this language as part of it. Many laboratories would use very similar language.
What is item 9-32.2.1?
Sample from stain A of the exterior left pant leg of the Tyvek suit from bag number 6 at 300 4th Street.
And is this the same Tyvek suit that we just discussed previously in 9-32.1.1?
Yes.
And what was the DNA profile result for this item?
For this item, there was the DNA profile was a female DNA profile originating from a single contributor.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
Yes, the DNA profile is at least 30 nonillion times more likely if it originated from Anna Walshe than if it originated from an unknown, unrelated individual, and this provides support for an inclusion.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Brian Walshe is excluded from this single source female DNA profile.
What does that mean?
So the single source DNA profile is probably the easiest ones for us to do a comparison because we should be able to see if the single source profile we have from the evidence matches the one profile we have from our known standard or the reference we have. So when I say somebody's excluded, it means that the 2 profiles don't match each other.
What is item 9-32.5.1?
And that is a sample from stain D on the interior right sleeve of the Tyvek suit.
And is this the same Tyvek suit as discussed with items 3 and 4 that we previously discussed?
Yes.
But is this a different stain?
Yes.
And what was the DNA profile result for this item?
The DNA profile was interpreted as a mixture of 2 contributors, including male DNA.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
The DNA profile from this item is at least 110 sextillion times more likely if it originated from Anna Walshe and an unknown individual than if it originated from 2 unknown unrelated individuals, and this provides support for an inclusion.
And what is sextillion?
A sextillion is 1 followed by 21 zeros.
I'm sorry.
21 zeros.
Thank you. Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Yes, the DNA profile from this item is estimated to be 6 times more likely if it originated from 2 unrelated individuals than if it originated from Brian Walshe and an unknown individual. This provides limited support for an exclusion. Similar to before, the MSPCL DNA unit validation studies have shown that exclusions for the inverse likelihood ratio range of 2 to 1,000 have shown known contributors could be excluded, and adventitious support could be attributed to this conclusion.
So what is limited support for exclusion?
So it's very similar to the inclusion one I described in our validation studies. We also looked at profiles where since we knew who those mixtures were made of, those individuals, we would compare this to those individuals. And when there's limited data available, again, for comparison for one or more of the contributors in a mixture profile, the statistic would be lower, but it's on the exclusion side. And when it is a lower statistic on the exclusion side, we would like to report it as a number, just so we can be clear that there's a possibility that somebody who is truly meant to be in this mixture would be actually excluded from it because of there being limited data for support. So in this case, there is support for an exclusion, but there is only 6 times more likely for that support. The likelihood ratio is in that range of 2 to 1,000.
13.1.
This was an unknown tissue that recovered from the Swampscott dumpster.
And what was the DNA profile result for this item?
This was a female DNA profile originating from a single contributor.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
The DNA profile from this item is at least 30 nonillion times more likely if it originated from Anna Walshe than if it originated from an unknown, unrelated individual, and this provides support for an inclusion.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Brian Walshe is excluded from this DNA profile.
So I want to move on now to the next round of DNA testing. What is item 5-1.1?
This is a sample from swabs of red-brown stain K on a basement floor in 516 Chief Justice Cushing Highway.
And what was the DNA profile result for this item?
This DNA profile was interpreted as a mixture of two contributors, including male DNA.
Were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
The DNA profile from this item is at least 15 nonillion times more likely if it originated from Anna Walshe and an unknown individual than if it originated from two unknown, unrelated individuals. And again, this provides support for inclusion of Anna Walshe to this DNA profile.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Brian Walshe is excluded from this DNA profile.
What is item 7-10.3.1?
This was a sample of stained area C on the blade of a hacksaw, placard number 8, 300 Forest Street.
And what was the DNA profile result for this item?
For this item, a female DNA profile was obtained originating from a single contributor.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
The DNA profile from this item is at least 30 nonillion times more likely if it originated from Anna Walshe than if it originated from an unknown, unrelated individual, and this provides support for inclusion of Anna Walshe to this DNA profile.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Brian Walshe is excluded from this DNA profile.
What is item 7-10.4.1?
That is a sample of the handle of the hacksaw, placard number 8 at 300 Forest Street.
And what was the DNA profile result for this item?
The female DNA profile was obtained originating from a single contributor.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
The DNA profile from this item is at least 30 nonillion times more likely if it originated from Anna Walshe than from an unknown, unrelated individual, and this provides support that Anna Walshe is included in this DNA profile.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Brian Walshe is excluded from this DNA profile.
If I may have one moment, Your Honor.
You may.
What is item 9-22.1.1?
That is sample from stain A on a white towel, number 1, with red-brown stains on it that was found in bag number 2 at 300 Forest Street.
And what was the DNA profile result for this item?
There was a female DNA profile observed originating from a single contributor.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walsh?
The DNA profile from this item is at least 30 nonillion times more likely if it originated from Anna Walsh than from an unknown, unrelated individual. This provides support for inclusion of Anna Walsh to this DNA profile.
And were you able to come to a conclusion about the comparison to the known profile of Brian Walsh?
Brian Walsh is excluded from this DNA profile.
What is item 9-28.1.1?
This was a sample of stained area A on apparent hairs found in bag number 4 at 300 Forest Street.
What was the DNA profile result for this item?
There was a female DNA profile observed on this item originating from a single contributor.
Were you able to come to a conclusion about the comparison to the known profile of Anna Walsh?
The DNA profile is at least 30 nonillion times more likely if it originated from Anna Walsh than if it originated from an unknown, unrelated individual. This provides support for inclusion of Anna Walsh to this DNA profile.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walsh?
Brian Walsh is excluded from this DNA profile.
What is item 9-31.1?
That is a sample from cutting of red-brown stain on a piece of rug from bag number 5 at 300 Forest Street.
And what was the DNA profile result for this item?
There was a female DNA profile observed originating from a single contributor.
Were you able to come to a conclusion about the comparison to the known profile of Anna Walsh?
The DNA profile from this item is at least 30 nonillion times more likely if it originated from Anna Walsh than if it originated from an unknown, unrelated individual. This provides support for inclusion of Anna Walsh to this DNA profile.
And were you able to come to a conclusion about the comparison to the known profile of Brian Walsh?
Brian Walsh is excluded from this DNA profile.
Moving on to the third round of DNA testing. What is item 7-13.1?
It is a sample from area A on the head of a hatchet, placard number 8 at 300 Forest Street.
And what was the DNA profile result from this item?
There was a female DNA profile observed originating from a single contributor.
Were you able to come to a conclusion about the comparison to the known profile of Anna Walsh?
The DNA profile from this item is at least 33 nonillion times more likely if it originated from Anna Walsh than if it originated from an unknown, unrelated individual, and this provides support for an inclusion of Anna Walsh to this DNA profile.
Were you able to come to a conclusion about the comparison to the known profile of Brian Walsh?
Brian Walsh is excluded from this DNA profile.
What is item 7-13.2.1?
That is the sample from the handle of the hatchet, placard number 8 at 300 Forest Street.
And what was the DNA profile result for this item?
The DNA profile from this item was interpreted as a mixture of 2 contributors. Due to limited information available, contributor 2 was deemed not suitable for comparison purposes.
So, were you able to come to a conclusion about the comparison to the known profile of Anna Walsh?
Yes, the DNA profile from this item is at least 2.6 octillion times more likely if it originated from Anna Walsh and an unknown individual than if it originated from 2 unrelated individuals, and this provides support for the inclusion of Anna Walsh to this profile.
And what does it mean due to limited information? Contributor 2 is not suitable for comparison?
It just means that in this mixture DNA profile, the data that is observed for one of these two contributors is so low level that we would not be able to make a comparison to any known reference standards. So we test 26 locations. We require in our DNA processing that data must be observed at a minimum of 3 locations for us to be able to do any comparisons to it. In this case, data was observed at less than 3 locations for this second contributor.
And what is an octillion?
An octillion is 1 followed by 27 zeros.
And you said that you were able to provide support for Anna Walshe, but were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
Brian Walshe is excluded from the first contributor that's suitable for comparison in this DNA profile.
So what does that mean?
So since it's a mixture of 2 contributors and I can only compare one of the 2, that one contributor, the DNA profile shows that it's the likelihood ratio shows support for inclusion of Anna Walshe to this profile, but Brian Walshe is excluded from that suitable for comparison first contributor in this profile.
What was item 9-20.1?
That was sample from stain A on tape with apparent gauze with red-brown stains on it, found in bag number 2 at 300 Forest Street.
And what was the DNA profile result for this item?
There was a male DNA profile obtained on this item originating from a single contributor.
And were you able to come to a conclusion about the comparison to the known profile of Anna Walshe?
Anna Walshe was excluded from this DNA profile.
And were you able to come to a conclusion about the comparison to the known profile of Brian Walshe?
The DNA profile is at least 23 octillion times more likely if it originated from Brian Walshe than if it originated from an unknown, unrelated individual, and this provides support that Brian Walshe is included as a contributor to this DNA profile.
And what is an octillion?
An octillion is 1 followed by 27 zeros.
I might have one moment, Your Honor.
Yes. Cross.
Good afternoon.
Good afternoon.
I think you said it in your in some of your answers in regards to the first round of testing that you testified to. And was that testing, was the report issued on January 13th of 2023?
Yes, that's correct.
And in the context of some of your answers to the testing results that you told this jury about, for instance, sample 9-27.2.1 at paragraph 1 on page 2, you indicate that it was interpreted as a mixture of 3 contributors.
That's correct.
Now, as you were explaining that and some of the other conclusions and the results of testing, you used the phrase, at least 3 people, correct?
That's correct.
So when you write this report and you include the phrase, the DNA profile was interpreted as a mixture of 3 contributors, it's more accurate to say the DNA profile was interpreted as a mixture of at least 3 contributors?
No, when we are doing our testing, we are using the information that we have in our profile. And in order to do any comparison analysis or further any kind of interpretation on it, we have to determine the number of contributors. So in this case, the data that I observed showed that there was 3 individuals in this. There could be more, but it's not in the data that I observed.
There could be more, though.
Yes, obviously the instrumentation we use, there could be contributors that are there that are not detected, but the ones that I observed, the profile showed a mixture of 3 contributors, which is how I interpreted this profile.
And when you interpret the number of mixtures, like you've just explained to the jury, am I right in saying that since we each inherit one allele from our father at a particular locus and one allele from our mother, then we typically have 2 different alleles at that one particular locus. Is that fair to say?
We could have 1 or 2 alleles at a locus, depends on if you inherit the same characteristic from both the mother and the father, or you inherit 2 different characteristics from the mother and the father.
And when you inherit the same allele, your father gives you the same allele as your mother, that would be a homozygote. That would be another way to frame that.
That's correct. That's a scientific term for that.
As opposed to 2 different alleles, your mother gave you one allele, your father gave you a different allele, then you'd be considered a heterozygote at that locus.
That's correct.
So what you're doing is, and please correct me if I'm wrong, what you're doing is you're looking at all the alleles that you have in front of you on a particular sample, and you're trying to determine how many, and once again, correct me if I'm wrong, you're trying to determine how many peaks you have at a particular locus. And for instance, if you had, 2 heterozygotes combined in a 2-part mixture, you would probably expect to see 4 different alleles there?
If we were looking at a mixture of 2 contributors, we would expect to see up 3 or 4 characteristics at the location. When we are looking at the DNA profile, he mentioned the word peaks. They kind of look like an EKG. If you can imagine like peaks and valleys on an EKG, that's what our data looks like. And so when we're looking at that, we are trying to just not only look at how many of those peaks are present, but what the level of each of those peaks is, because that can also tell us if it's coming from different individuals. So you would expect that if the same individual is contributing DNA to it, at all of these 26 locations, they would contribute similar amounts of DNA at all the 26 locations. And so if we see some high peaks in one location and low peaks in a different one, that could also tell us that there is a mixture present as well. So not only looking at the number of peaks that are present, but also the level of these peaks.
And so there are some assumptions being made about the way you interpret the data.
The assumptions we're making is based in science that we inherit half our DNA from our mother and half our DNA from our father. So we expect to see 1 or 2 alleles or peaks at each of these locations. So we're using that information to then assess the profile to see how many contributors are in this profile.
And without complicating this too much, and I don't intend to do that, but there is observed in DNA testing data, not just at a particular allele, 1 allele if it's a homozygote or 2 alleles if it's a heterozygote, but there's actually times when there's a triallelic allele, all right?
That's correct.
And so that's all I meant about the assumptions. And I understand they're scientifically based, but there are assumptions being made, correct?
Right. So if there is something like a triallelic pattern, there is a pattern to them, and we do know what those patterns look like. And what triallelic is that during the cell formation that happens, there's a mutation that occurs in the DNA that causes an extra copy to be present, either of either the allele from your mother, or the allele from your father, or a form of that allele. And that would cause, instead of seeing 1 or 2 peaks, we would see 3 peaks present for that person. But those usually have a pattern to them, and we can look at that, especially in single source profiles, and determine that. I did not observe any such things in the profiles that I saw in this testing that we did.
It's very clear, isn't it, that you have in a lot of these samples, if not most of these samples, you have concluded that a profile that was associated with Anna Walshe from a known sample, you could see the same profile in many of these samples, correct?
For any of the ones that were single source contributors, meaning there's only one person, that's where I can make that exact matching from the reference.
And then the ones that are mixtures?
The ones that are mixtures, those we would have to compare the whole profile as a whole. So it wouldn't always just be a matching of the two different peaks that we see. It would be a matching of looking at that plus the information we have on the levels of that contribution to see how likely it is to observe that profile if they were included in this mixture versus not included in this mixture.
Thank you. We appreciate your very clear answers. And what I would like to ask you now is something that you said early on direct examination. Your DNA testing and your results that you're reporting to this jury, it does not inform you of how a particular biological substance or a nuclear cell that resulted in the DNA profile, how it was deposited on that particular object that appears here in the reports, correct?
That's correct. We cannot determine how the DNA was deposited.
And if I were to tell you and suggest to you that for many of these items that were tested, if they were found combined with other items in a bag, and those other items came into contact with each other, you would agree with me that you can't tell this jury if the biological substance that was tested on one object might not have been transferred simply from another object that it came into contact with in that bag, correct?
Overruled.
That's correct. It could be transferred from one item to another item in a bag.
And would that transfer, that word you just used, would that transfer also occur if two items or more were placed in a single bag and then that bag was actually compressed by a compactor that compressed that bag very tightly to reduce its size? Would it be even more likely that there would be a, quote, transfer between those two objects of biological material?
Objection to the hypotheticals? Rephrase your question.
Would transfer be more likely if two or more objects were placed in a bag, placed after that into a compactor, and compressed together where the items within that bag were compressed together?
Objection. Same question. Same result.
If two items come into contact with each other, there's a possibility of transfer of biological material on those two items, correct?
That's correct.
And does the possibility of transfer between two items, would it increase if those items are pressed together tightly for a period of time?
I'm not sure if the chances would increase or decrease if they're pressed together, but if two items were to come in contact with each other, there would be a likely transfer of DNA.
So, based on all of your based on all of your observations of the data and the results here, once again, you can't say how any of the biological material was deposited on any of these items, correct?
That's correct.
And you cannot tell the jury, and it's the limitation of the DNA testing, you can't tell when it was deposited, correct?
That's correct.
And for instance, if there is a particular object like a tool, let's say a hatchet, you cannot tell from your DNA testing if that hatchet actually ever came into contact with the biological material.
Objection.
I didn't finish, but maybe I'll start over again. Let me start over again. The DNA testing on a particular object does not tell you that that object was ever used against a person whose DNA profile appears on that object.
Objection.
Sustained.
Well, you have told us that the biological substance that was tested does not tell you anything about how that biological substance got on that item, correct?
That's correct. The DNA testing that we do cannot tell us how or when the DNA was deposited on an item.
And I think you told us this on direct, but I just want to make sure I understood it completely. The DNA testing that you conducted here on the biological samples, that can result, the DNA profile can result from any nucleated cell, correct?
That's correct.
And so we heard you refer to skin cells, correct?
That's correct.
And we heard you refer to blood cells, correct?
Yes, except for red blood cells, all other types of blood cells do have nucleuses.
Okay. And so when you are given samples to test, you do not determine in your laboratory, and you can't determine based on your DNA results, whether your DNA results are the result of a profile coming from a skin cell or a blood cell or some other nucleated cell, correct?
That's correct. We would not be able to determine the cell type from which that DNA profile was obtained.
And even though sometimes you might get a sample that's identified as a red-brown stain on an item, especially an item of clothing, even if you got a full profile from that particular stain, you still don't know if the profile you got came from epithelial cells or skin cells versus blood cells, correct?
That's correct. We would not know if it came from any blood cells that were present on that item or skin cells that might have been present on that item as well.
And on sample 7-13.2.1, I think that's the sample that you explained to the jury that it was a mixture of two contributors, correct?
That's correct.
And even though you concluded that it was a mixture of two contributors, you said that due to the limited information, the contributor two, and you put in, you actually referred to it as contributors, and you put the S at the end of contributor in parentheses, contributors, two is not suitable for comparison, correct?
So a contributor is just, it's just one contributor. Contributor two is not suitable. The reason there's a parentheses S there is because we use similar conclusions when we have multiple contributors that may not be suitable for comparison. The wording that we use, like I said, it's from our protocols that is designed to sort of kind of cover all different types of DNA profiles that we can see. So in similar conclusions can be written where we would say contributors 2 and 3 would not be suitable for comparison, which is why there is a parentheses S there. In this case, the only contributor that I'm talking about that is not suitable for comparison is contributor 2.
That's all I have. Thank you very much.
Redirect.
Nothing, thank you.
You may step down.
Thank you.
See you at the side of the bench.